Hello, everyone. Today, it's my pleasure to introduce the study about comparative genomic analysis of Chinese Erwinia amylovora strains reveals genetic variations, plasmid diversity, and potential common ancestor. Fire blight is a highly destructive disease that impacts rosaceous plants on a global scale. It is caused by the bacterial pathogen Erwinia amylovora. The initial occurrence of fire blight in China was identified in Ili, Xinjiang in 2015 and damaged apple production in 2016 severely. Spread to the Korla region in 2017, causing significant damage to Korla fragrant pear, and subsequently spread to Gansu in 2020. At present, the whole genome of Erwinia amylovora has been sequenced from Americas, Europe, Africa, Oceania, and Asia, totaling 255 strains. Up to now, the whole genome sequencing and traceability analysis at the molecular level of E. amylovora in China have not been reported. In this study, we performed whole genome sequencing and annotation of 13 fire blight strains isolated from China, as well as strains from Kazakhstan and Kyrgyzstan. By understanding the relationships between Chinese strains and other strains, we compared them with the whole genomes of all uploaded E. amylovora strains worldwide. We further analyzed genome collinearity, sequence variations, compared to the model strain CFBP 1430. Finally, immature pear fruit was inoculated with each strain in this study to explore differences of pathogenicity. The complete genome sizes of the targeted strains vary from 3,798,299 to 3,805,877 BP. With a genome GC content of 53.6%. ANI, average nucleotide identity, analysis was employed to assess the nucleotide sequence similarity of the genomes, with similarities exceeding 95% classified as belonging to the same species. Genomic structural variations, including SNPs, single-nucleotide polymorphisms, and DIPs, short deletions, insertions, and other polymorphisms, were detected among all 15 sequenced strains. The results revealed a total of 5,087 SNPs and 1996 DIPs shared among the 15 strains. Besides, 172 common SNPs were identified among all strains, constituting approximately 50.7% of the total SNPs. In terms of DIPs, 28 shared sites were identified, representing approximately 21% of the total DIPs. Our phylogeny analysis showed that 13 sequenced Chinese strains were clustered in the same subbranch, suggesting a common origin for these strains. Furthermore, the Chinese strains showed the closest clustering with strains from Kazakhstan and Kyrgyzstan, which were also more closely related to strains from the Middle East, followed by strains from European regions. A further phylogenetic circular tree revealed the relationship of 15 E. amylovora strains in this study among with 255 E. amylovora strains from various parts of the globe. The results are consistent with the description above. The virulence assessments on immature pear fruits were conducted by 15 sequenced strains in this study. The virulence was quantitatively assessed by calculating the percentage of necrosis. While KL17-17 and KL18-6 exhibited weak pathogenicity with 0.58% and 4.75% necrosis respectively, 1,506-4 and 31,070-3 showed very strong pathogenicity with 67.31% and 88.43% necrosis respectively on 9 days after inoculation. To sum up, through full genome sequencing and comparative genomic analysis, all 15 strains exhibited a chromosome size of approximately 3.8 megabits. Population genomic analysis reveals that Chinese strains have a common ancestor and may originate from Central Asia. The speculated pathway of fire blight transmission to China was from Europe, the Middle East, Central Asia, Xinjiang. The 15 sequenced strains of E. amylovora displayed significant differences in pathogenicity. While KL17-17 exhibits weak pathogenicity, 31,070-3 shows strong pathogenicity.